Hello, everyone, and welcome to the Biotech Blueprint podcast. My name is Katarina Rosnick. I'm your host today, and our guest is my friend Patrick Creisher. Welcome, Patrick.
Hello, everyone. I'm super excited to be here today and
Very
and
excited
uh,
to have
chat
you.
with you.
Yeah, so Patrick holds a PharmD degree from Duquesne University and also a PhD from the Johns Hopkins University. We were actually in the same department during our PhDs, so that's how we met and became friends. I'm so glad to have you. And Patrick is an expert on animal research, and he did his PhD, and if I understand correctly, also your PharmD doing some mouse work and animal studies. And then you pivoted a little bit in your postdoc to organoids. So can you quickly introduce your PhD work, your postdoc work, especially in terms of animal studies versus organoids, and maybe also explain to our audience what are organoids and what exactly is your model organ?
Sure. So happy to chat. Um, you are correct that I have worked with we say animals, but it's primarily been mice.
Mhm.
The kind of gold standard for a lot of biomedical research in both my PharmD research, where I was studying the pathogenesis of measles virus infection using a mouse model, as well as during my PhD, where I uh, helped to develop novel mouse models of infection during pregnancy of various infections, including working with Zika virus, as well as influenza A virus. And then, I did my PhD as you know, and as you did, partially during the COVID-19 pandemic. And so we worked to develop a mouse model to help understand how SARS-CoV-2, the virus that causes COVID-19, um, has adverse effects during pregnancy. And now in my postdoc, I don't know if I would say necessarily a complete shift, but I did want to expand my toolkit to, as someone who is interested in staying in academia and having my own lab one day is, moving beyond just animal models and learning new skill sets and learning organoid models and as well as cell culture and some of these other skill sets. And so as a result, I have been working to develop novel organoid models and help to characterize them with other people in the lab.
Mhm.
And
Yeah.
so
In your PhD, when I was rotating in the lab, you were already working with like an organoid system, right? That was like a placenta.
So we were doing at the time we were doing mouse trophoblast culture, which is like a primary cell. So
Right.
I guess I can explain this for
Yeah,
the
what an
listeners.
organoid
Is
is. Yeah.
generally in terms of cell culture, and in terms of models for scientific studies as well as for drug testing and things like that. Things tend to follow a kind of like a hierarchy of where as you go up in the hierarchy, things become more complex, which adds additional variables, additional confounders, and things to your study, but does make it more physiologically relevant. So at the very bottom, you have cell lines, which are, taken from, they're isolated usually or generated from cells, and they are just in a dish. They're what you think of when you think of like cells in a dish. Isolated, they're often cancerous or have some other mutation that allows the cell to basically grow indefinitely. And so, with those kind of cell lines, they're more simplistic. We can sequence their genome so we can understand how they work biologically, but they're not necessarily relevant to, the human person,
Mhm.
because they're very simple. Then you can go up from that and do, so we consider that like 2D culture, with cell lines, and then we can move up, and another layer of complexity is what are called primary cells. So these are cells that have come straight from a person or an animal. In my case, we were trying to culture trophoblast cells, which are cells within the placenta, uh, from mice. So we took pregnant mice and tried to isolate their trophoblast. And it ended up not being useful for the project, so it didn't end up going anywhere, but it was actually what I was doing at the time. And so that's primary cells. They have the advantage that they're, straight out of a person or an animal, and so are more relevant to how those cells behave in the person, but because they are straight out of the person and they're not transformed, which is what we call it when you make a cell able to basically grow indefinitely, they often cannot grow indefinitely in your cell culture system, in your dish, in your lab. And so they often are limited by how long you can keep them alive. Um, so then another step up in complexity are what are called 3D cultures. So there's different types of 3D cultures. Some are called are like air liquid interface cultures, where it's like these are used for like respiratory infections to study like the lung or the airway, where you have cells that are like human nasal epithelial cells or cells from other parts of the respiratory tract. And then they're differentiated. And then on the bottom, you have media, which feeds them. That's like media is the
Yeah, like the milli liquid. What?
Yes, like it's like a liquid, yeah, that is used to
Yeah.
feed the cells. And then um, they're initially in what's called a trans well, where the cells are in between or kind of like squished in between two things. And they're in the middle, and then there's media on both sides. And then at a certain point in the process, you remove the upper media and let air, and that air causes the cells to think that they are like they would be in your lungs or your respiratory tract, and they become more lung-like.
So is
So
this
that
like an
is
organoid then? Would that be an organoid?
so it's kind of an interesting thing to ask is like the definition of an organoid, I think, depends on who you talk to and who you ask. It's not a hard and fast definition. I think some people would consider that an organoid. Um, but I think other people would not. So I think that for the purposes of our discussion today, I think you could probably lump them in with organoids as a form of a 3D, more physiologically relevant culture system. The thing that tends to make organoids is generally their ability to be passaged. And so that is it makes it so that way you can grow them basically indefinitely like you could a cell line. And so the reason for that is because organoids tend to be based on they come from stem cells of some capacity. So whether that be stem cells that are taken from a patient or from a donor, and those are like adult stem cells that are those are like adult stem cells that are usually, kind of differentiated, so they can usually only become a single cell type or
Mhm.
a single, like a class of cells. So, like, if you take stem cells from like the eye, for example, you can take like corneal stem cells or conjunctival stem cells from your eye, the outer portion of your eye, and then you can grow them and make organoids from them. But those stem cells will almost, unless you do something special, will usually only grow into organoids that are like the eye. So
Sure.
you really can't make a
Yeah.
adult stem cell from a specific niche into some other thing.
So
Um,
that kind of answers, I guess, one of my questions that I had, and I mentioned it to you before this, that when I was doing some of the research for this, it mentioned as a disadvantage of organoids that, they are like fetal because you have the stem cell, right? You usually do this in your lab, that you give them some factors, they develop into, like you said, the cell type of interest, and then they sort of resemble like a fetal neonatal stage, right? But you're saying that you can passage them, so you can grow them longer, so that then eventually they'll resemble an adult tissue,
so
right?
it really depends. So the thing about organoids is that's I think important to think about is that um, there are a lot of them, and there are a lot of them from different tissue types. There's a lot of different ways to grow them. So, like I just gave the example of adult
Mhm.
stem cell derived organoids. People will also derive organoids from embryonic stem cells that are more malleable and can become more different things. People can also do what's called an induced pluripotent stem cell, or iPSC, where they take like a fibroblast or a cell from a different type of tissue, and then they give it growth factors and cocktails, and modify it to revert to its more stem cell like state. And then from this stem cell like state, you can then grow them out to be lots of different cell types. But yes, in general, you can um, increase the maturity and the differentiation of a cell type by increasing um, how long it's cultured.
Yeah, so I mean, goes without saying why I brought you on this podcast about animal models and biomedical research. And so before we get into more of the regulatory stuff and your opinions on things that are supposed to replace animal research, I'll give some background for our listeners. So for decades, the US law required animal testing for new drugs largely in response to tragedies like the 1937 elixir sulfonilamide disaster, which killed over 100 people and led to the 1938 Food, Drug, and Cosmetic Act. And since then, drug developers have been required by law to conduct animal studies, usually in two species, often rodents or primates, before advancing to human trials. So preclinically, we have to have by law basically animal toxicity studies. But by the 2010s, this model was under pressure. Scientific advances and accumulating evidence show that traditional animal tests can sometimes fail to predict human response. And so in late 2022, a turning point came with the FDA Modernization Act 2.0, which officially ended the federal mandate for animal testing and allowed alternatives like organoids, which we just discussed a little bit, but also organs on chips and AI-based models to be used in preclinical testing. I'm assuming alongside animal studies, at least for now. And so now, as of April 10th, 2025, the FDA is taking this a little further. The agency plans to phase out animal testing for monoclonal antibodies and other therapies, favoring modern human-relevant tools, again, such as AI toxicity predictions, lab-grown human tissues, or real-world safety data from abroad, from other countries. And so companies using these approaches may now even qualify for accelerated reviews, which is something they want, obviously. And the FDA, new FDA Commissioner Martin Makary called it a paradigm shift, one that could cut drug development costs, speed up timelines, and spare thousands of animals each year. Just as a side note, do you know how many mice and rats are sacrificed annually in the US alone in research? Wild
No,
guess.
not, I mean, it's probably a big number, but I don't know.
Yeah, it's over 110 million. So it's a big number.
Wow.
There's this ethical
Wow.
also.
Do you know, is that like just in the as part of the regulatory process, or does that include like just like basic science research labs as well?
It's all in all.
I see.
But I mean, it's a lot. It's a huge number, and you know that's like this ethical side of it that we might not be able, may not have time to touch on. But it's a huge number for sure. But so my question for you now is, do you think that companies that breed mice for research and you know supply research animals to institutions, to companies like Jackson Labs, like Charles River Labs are going to struggle with after this unless they pivot? For example, the stock of Charles River Labs really plummeted after April 10th, and then they sort of said they issued a statement that they're going to try to you know, adapt and the stock recovered a bit. But do you think the sentiment is a little premature as of right now, or do you think this is where we headed?
I think that's a really interesting question because I think a lot of people who work with animals in research are always trying to reduce the number of animals, and you mentioned the thing about ethics and trying to work with animals in the most humane way possible. So, like the FDA has followed the what's called the 3R framework, which is replacement, reduction, and refinement, which have been, I think they were developed like over 50 years ago to provide a framework for more humane animal research. And um, this is kind of just the next step in that kind of process of trying to reduce the number of animals that are used in research. And I think an important thing to think about as well when you mention like these companies and the stocks is to, there is more to research than just, the regulatory process and, just drug testing. So like, for example, like Charles River, Jackson Labs, they provide animals for and mice specifically for
Research.
studies, that are done in like academia and industry that maybe don't directly lead to the approval of a new compound or something. And
Sure,
so
sure.
they're
But I
used
think,
for general uh,
yeah.
increasing
But
our
I mean,
knowledge.
I think that if, you know, the FDA basically is telling companies, you know, try not to rely as much or not rely, that's maybe premature, but try to come up with alternatives, right, to animal research. And I think that academia might be slower maybe to adapt these things, but I don't know. I think that they might follow, eventually. And so, um, I actually wanted to ask you, like, one of my questions was, do you think that it's going to be increasingly difficult to maybe get funding for studies that solely rely on animals?
I think that I think a key point to think about, because I agree that like I think as the landscape shifts, I think things will have to change. But I think a key thing to think about will be like trying to not look at the whole system with it kind of from a broad perspective and thinking about, you know, I think that there are, and kind of thinking about it at a more case by case, kind of situation by situation, field by field basis. So I can think that there are probably studies that can be done with the way the technologies are now, where animals are not required. And I think a good example of that is also mentioned in the FDA roadmap is like things where a compound is approved in Europe or in a different part of the world, and they have data that has not been, you know, vetted or analyzed by the FDA. And so, like, utilizing that data and following the roadmap, so that way, data that's currently available from studies in humans can be used. But I think that there are studies where, you know, there's it could be a complex system where the technology with organoids, or other like the organ on the chip or other models is not there yet, and so like maybe we'll get there one day, but I think there will be complex systems that are difficult to study in their full capacity in vitro systems. It's kind of interesting because I think like depending on who you ask, you'll get people that come at it from different perspectives. So like you'll hear someone who's like, oh, if it wasn't, it's only done in an organoid or done in vitro. So like, is it really relevant to humans because we're more complex? So like you have to test it in a mouse. But then you'll have other people who will be like, oh, well, a mouse isn't a human, which is true.
Mhm.
So you need to test it in an organoid. And I think an important part of this process and thinking about this is, you know, trying to think about the regulatory system overall and how, what do we define as safe? Um, what kind of overall systems do we think about, and like where do we reach the bar for what is okay to give to people? You mentioned, I think earlier, if you want to talk a little bit about it, the some of your thoughts on like organs on chips and across different systems, and like where do you find that line? And I
Yeah.
think the thing you said was interesting. So
Yeah, yeah, no.
uh,
So yeah. Actually, the next question I have is because the the FDA really they outlined three alternatives, right? Like you said, there are also other things, but the main three things they outlined were organoids, which we discussed a bit, uh, organs on chips, which for I'll put that into the Substack article that I'll write with this podcast, which is basically like a USB stick size miniature organ as of right now, at least. There's like lung on chip or heart on chip, and it's my understanding is that it's a little bit more advanced than organoids. Um, you can have there's a company now out of Germany. They're called tissues. Um, and they've created more like a 2 organ, 4 organ chip system. So you can simulate more of a human body, I guess, in a better way to see how a drug is metabolized or how, treatment or anything can really affect different organs. But we were talking a little bit about this before, that the question really that arises is how many chips representing how many organs do you need to cover all critical systems, right? Like if you test the drug or anything else on a liver, heart, and a kidney, a chip that's a multi-organ chip, and then you see no issues, is that considered safe? Can you now be like, okay, it's safe, I can test it in humans now? Or, perhaps not entirely because you maybe didn't include other systems like nervous system or, we talked about immune system. It's not really a part of, immune cells are not part of the organs and chip, which also brings an issue, or you don't have blood vessels there, right? So, I mean, I can see a future when maybe these are much more sophisticated, right? And like you could have like a miniature human like system maybe. But it's definitely quite I mean, yeah, definitely in the future. I don't know how many years, like I don't know this would take, but yeah, what do you think? Like, do you have any?
Yeah, so it's interesting that you mentioned that you th would consider organ on a chip like quote unquote more advanced
I don't
than
know,
an
is it?
organoid. I would say that they both have their like pros and cons. So the thing that to my understanding, which I will say, I don't work on organs on a chip, but from my understanding, one of the things that makes them unique is that they use like microfluidic systems, so that way they can communicate
Mhm.
um, with each other, and so that's really, I think, one of the main drivers. And I think organs on chips are great for screening purposes, and but I think that in being able to, so we mentioned earlier, I guess I'll say this to get into that to kind of transition a little bit. We mentioned earlier about like media and like the media to support cells. And so like one thing to think about is that different cells, different organoids may need different medias. And so oftentimes these microfluidic systems can allow for communication between cell types that, if you put them in one dish, you couldn't probably get them to grow together well, because they require different environments, different signals. And so that's really, I think, one of the advantages of them. Um, but
I don't
yeah,
really know, like, I honestly, like, I've heard of it about it, obviously, like in my PhD, but I don't know if anyone who does that kind of work. And like, is it pretty new? Like, do you know, like, do you know if anyone who works on organs on chips, or like in basic research at least, or like in like academia, like, is it pretty common to use that kind of technology?
I don't know if I feel I don't if I have the qualifications or knowledge to say common or not, but I mean I've been to a few talks where I've like, seen it.
Yeah.
And I think we're in the stage currently where there's a lot of like validation, like
Mhm.
studies that are like, look, we're trying to compare this system that we developed back to tissues from humans. And I think that you have to do all of that before you can use these for, applications like studies and stuff. And so oftentimes the papers that are in that that these that are published with these systems are like, they develop the system, and then at the end they say like, here's one potential application, but
Mhm.
that's just one application, and all of these tools and models can be used for lots of things. And so I think as people become more comfortable, the technology evolves, and also not just evolves but becomes more inexpensive,
Mhm.
right? So like if a system is something where you need to have this complex system built in your lab that only one person at the whole university can has, then like then there may be some logistical issues and things. But
Also, it's very
as
technical.
things develop, get smaller, things can potentially be like 3D printed. Like I've heard of people potentially like 3D printing some of the components of these systems, so that way to like drive down cost and things like that. um,
Yeah, I mean, I don't know the price on that, but I can imagine it being expensive, and plus like a disadvantage, I guess I can think of is the technical skill, right, that you need to operate these. I assume. I mean, they're like little, systems that I wouldn't even know where to start. A tiny, like, what do you do with that? How do you keep them alive and happy, the cells? And as opposed to like mouse, rodent area where you have like a technician who can help you out a little bit, but here you kind of need like that high level skill to operate
I
that.
know that at some institutions, I can't think of any off the top of my head, but I have seen that like cores are being developed in that vein to help investigators in that capacity. And
Yeah.
I think that if this system that, this regulatory system that we're talking about today, you know, as it comes into being and as more companies go through with it, and therefore more basic science needs to go into this vein, then I think then that will probably have to become more and more common, because like you said, there is I think a lot of technical skill, which I think is useful for, expanding the technology to people who may not have those skills but have ideas or have other systems that could be combined with them. I think that's where these kind of cores. Do you want to explain what a university core
I was just
is?
gonna say that. Yeah, yeah,
Because maybe you can
yes,
explain that because the
yeah.
listeners may not have experience in
Yeah,
those kind of
yeah.
things.
So a core is, at least in academia, it's a part of institution. It's like think of an office where you have specialists for a specific technology that we use as scientists. So let's say we use a machine that's expensive. It's hard to get people trained on it. And so while we as PhD students or PhDs might be expert on, like, we can use it, it's always good to have someone else to basically help you, you know, service the machine, start the machine, help you analyze some of the data that may come out of it. And so we have these cores that specialize in things like flow cytometry or I don't know, sequencing
Sequencing, I think that's a good example.
and so Patrick is saying that he has heard of a core where they could maybe potentially have some of these organs and chips or organoid like.
Organoids too, yeah.
Yeah, that is interesting. Yeah, I didn't know that. So yeah, so I think
So yeah, I mean, I think in that vein, I think so. Part of, I guess we can continue that train a little bit in the sense that like one of the things with organoids, organs on chips, and a lot of these systems are that it is often a high barrier to entry to get these systems set up. So, like for example, if your organoid is based on patient samples or donor samples, right? You need to get those from, you need some kind of agreement with the hospital or with a company or something to get those to set up your system. Um, you need potentially like a microfluidics platform of some capacity to do the organ on a chip.
yeah,
And there are companies that provide those kind of things, but I think it's not as simple as like, a basic cell culture where you can just order the cells, put them in a dish, and
definitely.
and I think that's an area where, animal research, because animal research and rodent research has really been a mainstay of biomedical research and for a long time, is that the logistics, the systems are in place, and
Yeah.
so I think any kind of shift in the process will need accompanying shifts in the logistics and the systems of support, because otherwise I think that you run into issues where things are just not feasible for most people or for new companies or even established companies that have always been doing something a certain way.
Mhm. Yeah. No, that's a good point. And I mean, right, the regulatory acceptance, I think, of organoids and organs and chips is definitely still in like early days, right? Like we don't have any drugs right now accepted solely on data from any of these. And I think the science in general, it's maybe not early days, maybe that's maybe a little exaggerating, but like earlier, right? And I think the regulators have shown interest. We will have to probably, like you said, or we've hinted at this, do both things side by side, validate them, right? And then maybe eventually there could be a future where we solely rely on them, but who knows, right? It's definitely more of an earlier um, technology that is being adapted, maybe, and maybe this will happen first at the companies, and it can maybe trickle down a little bit into industry, into academia, but yeah, we'll see.
Yeah, I think an important part is that, like, I think in all of these things is like keeping an eye on the nuances
Mm,
and keeping an eye, because I think that there are systems that are more adaptable to these kind of things and systems that are less. So, like you mentioned, the immune system. And so, like, one of the things that the FDA started with and that they mentioned is like monoclonal antibodies. And so, those can be kind of tricky because oftentimes, depending on how they're made, they may have portions that are from human, maybe entirely human, may have portions from different animals. And so putting them in a mouse, it may react to a human portion, that a human wouldn't react to, or it could not react to something that maybe a human would react to. So I think that's an interesting part in terms of testing safety.
Yeah.
Um, one of the things to think about also is not just safety, but efficacy. So, like, if a drug potentially has a mechanism that requires like activation of a complete organ system, or like a pathway that is more advanced than just a single cell. So, like, thinking about like some of the like the HPA axis, for example, which is the hypothalamus pituitary axis, I
Mhm.
think. We should
Yeah,
we
yeah, I
should
know
look
what
that
you're
up.
talking
I think so. So like that's a system where it requires communication between multiple different organ systems.
about.
And so to test efficacy of something blocking that pathway, you would need a more complex system where you could recreate that communication. And so like if you could do it in like an organ on a chip because it's all like fluidic space, then like that could work. Yes.
Do you like, for example, if you're testing something that affects the brain, like you said, or like the glands that you mentioned, do you know like is that stuff done now in like primates, or do they do that in mice, and or how do you know of any, or was that just an example?
That was just like an example. I mean, I don't know off the top of my head any specific examples from like the regulatory side. I know that a lot of times in the basic science side, a lot of things are tested first in mice because they are less expensive, and
Sure.
therefore can be
Sure.
used to test early hypotheses versus, the people who work with non-human primates who do those kind of studies. It's more limited, tends to be more expensive, and so people will move there after they have, a potential
Sure.
uh, positive result in a mouse. That doesn't mean, I think the FDA cites in their reasoning, of course, everyone knows that there are examples of things that worked in mice but didn't work in people, or things that,
Yeah, I
worked in
actually
people but
saw
didn't work in mice. So,
that it's like 90%. Do you agree with that?
yeah. I don't know. I'm no expert on those kind of statistics. So
Yeah.
I haven't had a chance to look into the source. But I mean, I think a lot of important research has been done in mice. I think a lot of important research has been done in cell lines, before we had organoids. So I think it does a disservice to all kinds of science,
Yeah.
regulatory science, to paint these kind of things with like broad brushes, because there will always be systems where things work better for some things and not for others. I think, you mentioned earlier that like AI, and maybe this is a good point to chat about that, is because there may be systems where AI may be more beneficial and systems where AI may be less beneficial. I know you mentioned some companies that are doing some stuff with AI. Maybe you want to mention that.
Yeah, yeah. I think for this specific use case, when you have especially like replacement of toxicity studies, right? Like that's the main thing that concerns biotech and pharma industries. You have basically, I think personally that AI can be helpful here. I mean, we've seen it with companies like In Silico Medicine, which is I think a company based out of Boston, and they are one of the first companies that actually were able to find a drug candidate and predict a safety profile before going into human trials, obviously. And they didn't, I mean, they didn't do it solely using AI. Then they basically selected a few candidates, is what my understanding is. And then they did some limited animal research, but ultimately what it did was, and it's a fibrosis drug. It's not cystic fibrosis, but it's like a type of fibrosis in humans. And so they were able to basically do this in a fraction of the typical development time. And also it was, I don't know about cheaper because, this compute power that we need for AI, at least right now, I'm assuming it's quite expensive, but maybe cheaper than the traditional research. Um, and so it cut down drastically the number, the time needed, and then the number of candidates that they selected by basically filtering compounds virtual. And you're the PharmD, so I don't know how they did it. I guess they had like a toxicity motifs that they tested and excluded companies compounds based on that, is my understanding. And they have a really cool pipeline. So if you look in on their website, they have, I don't know, 20 drugs like that. They're all AI generated. They're all screened in animals. So, kind of goes hand in hand. You can't just replace AI or animal testing with AI. And then they basically advance it into human trials, and they all seem to be doing fine. Like they're safe. Yeah, it's kind of cool example, I think. Um,
Yeah, so I mean, I think that is an area where there is potential because it's something like, saving time for something that a human would do. Like you mentioned, there are like known motifs. It's often referred to as structure-based drug design, right? Where there are like known motifs that are known to interact with certain receptors in the body, and so you can predict
a
that
motif being
structurally.
like a part
Uh,
of the compound, right? That
Yes,
is
exactly.
toxic, basically.
Right. Well, not even toxic, but just like, known to interact with a certain protein in the body. And, in some situations, maybe that's what you want, right? You want to be able to screen compounds for, let's say you have like a known protein that when bound, turns it off. And so if you have a disease where you want to bind to that protein and turn it off in
Mhm.
that situation, then, if you know what the structure of the molecules in a drug that could bind to that receptor or bind to that protein, then you could screen a bunch of known drug libraries for that. And basically cut down on the amount of time to find candidates to potentially do more extensive testing.
Mhm.
I think that's one of the areas where it could be, beneficial, and one of the things where I feel like, obviously AI in this context is very much in its infancy, like you said, but I think there is definitely some potential.
Yeah, and I think, this is not that sci-fi-ish because like I guess AI, right, it improves the more data you give it, the more it improves. And so you can think of, and I'm sure there is some limits right now as to the compute power and all the data you can give it. It's expensive, but you can, I guess, create like a large database of all trials is like what how I think about it, and like give it examples of toxic compounds, what worked, what didn't work, and basically, use machine learning to predict the likelihood of a new compound to, cause a liver injury, cause DNA damage, or be safe. The alternate, the desired outcome.
Yes, I agree. And I think that kind of hits on where some of the limitations are as well, wherein, it only knows as much as we have taught it.
Right.
And there will almost certainly be new compounds that have new reactions that we just don't know about them yet, in which case, it can't be predicted. But after we have, we learn something and we give it more information. So, teaching it from research studies, from clinical trials and stuff, which things that are,
Yeah,
continually ongoing, then you can, help to further improve the system
yeah.
to,
The clinical
yeah.
trial thing that you just mentioned, I also just was reading about basically, again, same thing, right? Like same idea. It's like the digital twin or virtual clinical trial thing where you basically give the algorithm as much data as you can from like successful clinical trials. Like it's hard for me to imagine. Like I can imagine the toxicity screening, like how you can find a or somewhat can imagine like how you find a compound that's not toxic and then you like test it in a lab. But the clinical trial is a little harder for me to understand. But like you basically again train the algorithm, give it as much information, as much virtual, like you basically create like a virtual patient population and again, like it's not to replace clinical trials, right? But maybe like a tool. That's kind of how I think about AI, I guess. At least right now, it's like a helpful tool to a scientist or whether it be in academia or in industry. And I think that it has a potential in that sense for sure. But of course, there are limitations. Um, like with the
Right. So I think a good example that you just made me think of is in the context of like toxicity screening, and like, so like uh one way to test for that is to basically, um take a bunch of compounds and basically put them on a bunch of different cells and like look at them and like, but as you increase the number of compounds, you can imagine that like that's more work to do. And so if you can use AI to reduce the number of compounds as a starting point and say like, hey, these ones are like very obviously going to be toxic. Uh,
Yeah.
and like maybe a person could have looked at the compound and said the same thing. But if you have like, 5,000 of them, I mean, that's a lot of time for a person to like, scroll through a list and be like,
Yeah,
versus,
definitely.
you could have a neural network or something like that, do that kind of screening
Yeah.
um, much quicker.
Yeah. No, for sure. I think if this in silico drug that they're testing is if it gets approved in a few years, I think that would be like a good proof of concept that you can do both. You can limit the number of animals, for example, that you need. You use it as a tool to help you streamline research. So I think it's cool. And like, what do you think? Like, do you think we have a future where we actually won't need animals to do research, or do you think that's like way in advance, or do you think that could be like 10 years from now? What do you
Oh,
think?
that's a tough question, I will say. I
Yeah,
feel like, I think one of the things is that like it will probably be like field context dependent. So I'm sure there
yeah,
may be things where, it can be phased out much more quickly than in other areas. Your training is in immunology, which I could imagine is a more complex field that may take longer to really phase out on these kind of testing, really even like um even using stuff with human samples and like those kind of things versus this kind of computational um
toxic.
information. I think at the end of the day there will probably always have to be some kind of validation. Like I can imagine, I think I mentioned this earlier, but like I think that the model will always only be as good as the information we can give it. And if there's something that is brand new, something that we have never encountered before, like that would be, in my opinion, very difficult to predict. And so I think
You
that,
mean completely new drug, basically, for example.
or like something that like, not even necessarily like a drug, but like a new interaction, like, maybe something, has it comes together such a specific way that it triggers an immune reaction that like we don't have compounds that have
Yeah,
uh
yeah.
act in that way or interact in that way, and
That's
so
like I'm
that
sure
would be
that
something that you would
yeah,
need to
valid.
worry about.
Yeah, yeah, definitely. I mean, like I think that again, this is gonna improve, right? Like the more data you give it, exactly like you said, the better it's gonna be. But definitely there's limitations, at least the way things are right now, right? We you don't want to risk that you move too fast with something unproven that can, be risky to human safety. So it's like another ethics viewpoint, I guess, for like the opposite one. Do you think that what is your like opinion on AI overall and just in general? Like also, do you use it? Do you guys talk about it like in the lab? Like, is there anything that's on your radar in academia or
So
in
I think that I think you mentioned it really well in earlier when you said that, it's like a tool. So
yeah,
I like to think about it in the same way as like, a tool in the context of like things like spell check, things like, things that make our lives easier. I think that, it's a little more complicated than that because of, the ethics of the environment, the environmental aspect of, you know, AI and the cooling of computer farms and things like that with like water and resources and things like that, which I think make it a little more complicated. In terms of like using it for example to like I know you hear stories of people being like, oh, I didn't want to Google something, so I like just asked an AI, in which case
Yeah.
or like, I mean, even when you Google things now, the
Yeah.
first option is like the AI thing. And so I
I
think
think that's very
it's a
common.
tool. I think it will be interesting to see like how much of it this is like a fad, right? Like I remember when I was growing up, and there was this idea that like there would be like the Google Glass, right? Like the glasses that like
Yeah.
would be connected and you could like see, and it was like going to come out and be like the coolest thing, and then like that was supposed to be the
Well,
future, and then
I'll say though that I went to Costco like 2 weeks ago, and there were people from I don't know what I think it's Google, and they had Google glasses, and man, they're cool. Like it looks like Ray-Ban glasses. You can't see anything on it. And like, I mean, obviously it's not like Googling things, but like
yes.
you can Google basic things. Like it's operated by voice. I put them on. I mean, they are light. They're cool. And like you can take pictures. And I thought that was pretty cool because like just a side note, because I'm like, I want those glasses because they're like $200, which is, a lot, but not as much as you would expect a glass like and so it takes really good pictures. And I'm like, hey, if I can, go do something and not have my phone in my hand, and can take pictures of my glasses, I mean, it's a cool thing. But definitely
Yeah, no, no,
they made it sound like it would be like everyone would
like
have.
everyone would have them, right? And so, but like obviously
Well, it's a
that's
good
not
point.
like the
Yeah.
case yet. But like I think it will be interesting to see. I'm sure that there, like we've talked about, that there will be uses where it will have a clear benefit,
Yeah.
um but there may be places where there may not be a benefit or may even be a harm, in which case I think we'll have to evaluate it on like a case by case basis. But I think that it will be really interesting to see as these models evolve, as things change, as things get more competitive or less competitive. I feel like nowadays I feel like everything is like has AI associated with it. Every
Yeah.
company's trying to like do
Yeah.
something with AI, and that may be not the best. May not be the best moving forward, but it will be interesting to see how
Yeah, I
these
think,
things progress into the future.
yeah, I mean, I see it too. Like, when you see all these like company accelerators and startup accelerators, everything funded is AI related, right? And at first I was like, oh my gosh, like everything is AI. But then when you think about it, I mean, some of these companies will inevitably be big because if AI is in the future, which I think it is in some ways at least. Also, speaking of a tool for scientists, I spoke to a founder of, and this maybe you could find it useful. I'm out of research for now, at least. So I don't really use it. I wouldn't use it, but like I would thought this was like I used it, like I tried it out. And like I think it would have been really useful for me doing my PhD. It's called, I'll send it to you. And I'm gonna probably like write an article on this too, but it's called Potato AI. And it's because like back like when you're a kid and you do these science experiments like the potato experiment, and so that's why they're called that. But they basically are like a so he's a scientist, I think neuroscientist from Brown, and then like turned entrepreneur, and he's doing basically like again like a helpful tool for scientists. And right now, at least the stages they're at is more geared toward academics. And like you should check it out. You can create a free account, and so it's like a second set of eyes. So I tried that, and like it gave me a really cool protocol to like use and try out, and it's like really good interface, like you can talk to it. And like I think it's like a cool way to again help you a little bit, because sometimes in your PhD like you're an expert on what you're doing, and oftentimes you're the biggest expert on this specific thing, right? And unless you talk to maybe people that are doing that somewhere else in the world, like you might be the only person in your like institution who's doing this exact thing. And so like their goal eventually is to roll this out to companies and like help you again streamline the way that experiments are done. But it's something to consider. Like you should look it up. It's really
Yeah,
cool.
no, that sounds really cool. I mean,
Yeah, yeah.
I know you did a lot of this stuff. I say back in the day, but I know that a lot of people um, utilize AI to help with like coding and like working like R and
Okay.
helping with
Yeah, like
troubleshooting
strategy is
scripts and
really good.
stuff.
Yeah. Yeah.
Yeah. And so I feel like that's just another example of it being like a useful tool.
Yeah, definitely. But yeah, any final thoughts that you have that you would like to share with our listeners?
No, I mean, it was great to thank you for having me on. It was
Yeah.
great to be here, chat with
Yeah,
you,
maybe
talk a little bit
maybe
about
we
my
can
experience. And
yeah, yeah.
yeah, maybe we can chat about something else in the future. I mean,
Yeah,
I
yeah,
love to
something
chat,
else
you know,
or follow
so
up on something similar. Yeah, that'd be
yeah.
great. Yeah. Okay, thanks, Patrick, and thanks
Oh
everyone.
yeah, no problem.
Yeah, thanks for listening.
Bye everyone.